Hello, my name is Mitch Kronenberg, and I'm at the La Jolla Institute for Allergy and Immunology. I'm actually the president and scientific director, but I have a very active research lab, and that's what we're going to be talking about, of course, is one of the technologies we have, which is CD1d tetramers. Most of the work in my lab concerns CD1d reactive T cells, and these cells are unique because they recognize glycolipids rather than peptides. And another branch of my lab is concerned with a completely different topic, the mucosal immune system and its regulation. CD1 molecules are antigen-presenting molecules, and that means that they bind antigens or fragments of proteins or other molecules and hold them up for T cell recognition. Now, there are really two different kinds of antigen-presenting molecules. There are ones that hold or present peptides, and there are ones that hold lipids and glycolipids. And the CD1 molecules hold lipids and glycolipids. That's their function. And they hold up or present these lipids and glycolipids for T cells. And there are actually four different kinds of CD1 molecules, CD1A, B, C, and D. And actually, we focus on CD1D. And CD1D in particular is probably the best studied, in part because they're mouse models. And CD1D presents glycolipids to a particular kind of T cell called a natural killer T cell, an unusual white blood cell that has features of the innate and adaptive immune response. The glycolipids that are presented can be self-glycolipids, although those are not very well characterized at this point, or they can be microbial glycolipids. And the glycolipids